Hello students, welcome. Form 3 topic 2, lesson 17. We are going to do volumetric analysis. So volumetric analysis is a method of quantitative chemical analysis in which the amount of substance is determined by measuring volumes of solutions. Usually, we are told two solutions are usually considered. One solution of known concentration and volume is reacted carefully with another unknown concentration to determine the reacting volumes. This data is used to determine the concentration of the said solution, and the above procedure is called titration. So we have a standard solution. So a standard solution is a solution whose exact concentration is known. Remember, we said two solutions are usually considered. One solution of known concentration. So that solution of known concentration is known as standard solution. So again, the solution whose concentration is unknown, it's called the analyte. Analyte. So during the exam, if you're told what is an analyte, you say is a solution whose concentration is unknown. Then we have the apparatus used in volumetric flask. I mean, in a volumetric analysis include or are pipette, burette. Apart from these two, we can have the conical flask. We can have conical flask for doing that titration. We can use again clamp and stand for holding that burette. Those are some of the apparatus that we are going to use when we are doing titration experiment. Okay, we got titration. Titration is a quantitative analysis process in which a solution of known concentration, that is the standard solution. This is standard solution, is added gradually to another solution of unknown concentration. So that is the analyte. That is the analyte. Because we said analyte is a solution whose concentration is unknown until the reaction between the two solutions is complete. So we have endpoint. What is endpoint? Endpoint is a point at which the reaction between the two solutions is complete, and we are told an indicator is used to identify the endpoint. Remember, in Form 1, we have done indicators. We were having the three commercial indicators. One of them was phenolphthalein indicator. We were having the methyl orange indicator, and we were having the litmus indicator. So if you are having acid, you know the color it will be having. Maybe phenolphthalein indicator in acid, it will be colorless. If you are dealing with a base, phenolphthalein indicator will be pink. If you are dealing with a neutral solution, that means phenolphthalein indicator will show colorless. If you are dealing with methyl orange and you are having acid, it will be pink. Base, it will be yellow. And also, that side of neutral, it will be orange. Then lastly, if you are using litmus solution, that means it will be red in acid, it will be blue in base, and it will be bubble in neutral solution. So, again, when you are dealing with the titration, there is a choice of indicator. Like if you are dealing with titration between strong acid and strong base, you can use any indicator. You can use any indicator. But if you are dealing with strong acid and weak base, you are supposed to use, or you will be better to use methyl orange. And if you are dealing with weak acid and strong base, phenolphthalein indicator will be the best suitable indicator to use. Then we are told titer. What is titer or titrant? Or titrate, I mean. So, titer is the volume of the solution that runs out of the burette in every titration experiment. That is called titer or titrate. Or you can say titrant, no problem. Then lastly, we are told usually 3 titration experiments are carried out. Then the titrate values of the 3 experiments should be consistent and have an accuracy of plus or minus 0.2 centimeter cube. Then next, we are going to go to types of titration. So in our Form 3 KLB, we are having 3 types of titration. One is direct titration, back titration, and also redox titration. So in our next class, we are going to start with the direct titration. That's the end of our video.